Hi, everyone. First, thanks to OCTS for inviting me to give this talk to you today. I wanted to do something a little bit different for the virtual platform and walk you from the bench to clinical development on one of our programs, show you the underlying technology, and then talk about how we move from bench to clinic to pipeline um, across our programs at Aleta. So, without without overstating it, CAR T cells, when they work well, can be quite extraordinary. And they work when they see and can bind to tumor antigens consistently in order to trigger cytotoxicity, serial killing, CAR T cell proliferation, all of which adds up to a persistent CAR T cell population that can outlast the tumor and eliminate the tumor for the benefit of the patient and providing that patient with a durable response. Now, as I said, when these work, they're amazing therapeutic modality. So the CAR T cells that have been approved are directed to the B cell malignancies in relapsed and refractory B cell leukemia and lymphoma. And I like this slide because it makes 2 points. One is that the therapy can be curative, and we see illustrated here a patient who's been on therapy 8 or 9 years. The other point that it makes is that these are patients for whom there was no other available care, and so you're really rescuing patients with curative effect, which is a remarkable achievement, and I think bodes well for the CAR T cell platform as it moves from the B cell malignancies into other tumor indications. Now, CARs that see CD19 are somewhat unusual. Their persistence properties is a natural attribute of their ability to see CD19 not only on the target tumor cell, but also on normal B cells. The normal B cell component of this is important because those CD19 positive normal B cells are a non-tumor dependent and self-renewing source of antigen, self-renewing from the bone marrow. It's an antigen depot that allows the CAR T cells to outlast and outlive the tumor cell population that they're eliminating. I'll show you a little bit of data that suggests that CAR T cell fitness is enhanced by interaction with B cells in a way that is not duplicated by interaction with solid tumor cells. And we'll talk later on about the trafficking properties of CAR T cells and how they can be used beneficially for the indication that we want to treat. So I want to leave this slide with 3 words that are important to us. The CAR T cells must have persistence properties, fitness properties, and trafficking properties that are beneficial. Now, CAR T cells to CD19 can work very, very well. Other CAR T cells just do not work as well. And the question really is, why not? Now, I think in the solid tumor space, we have a number of critical issues, and they're the same issues that are exemplary in CARs to CD19. CARs to solid tumors have poor persistence properties. Their fitness seems to be limited. They don't access tumor readily. In addition, solid tumors show rapid antigen escape, so they lose antigen expression in the face of cell therapeutic treatment. And we have to be a little bit careful about what antigens we go after because those antigens may be shared with normal cells and tissues that are critical, unlike B cells, which we can deplete without undue or unmanageable toxicity to the patient. So our technology has 2 key elements that we designed to tackle these critical issues, learning from CARs to CD19 and utilizing them as a platform therapeutic to go after other antigens. Why? Because those CARs to CD19 have the exemplary properties I just told you about. How we build bridging proteins that act as CAR T cell engagers to link those CARs to whatever antigens we wish to target on the targeted indication. So let's talk about bridging proteins envisioned as anti-CD19 CAR T cell engagers. They have to have a CD19 domain. That's the target for the anti-CD19 CAR. They can be single antigen binders, dual antigen binders, triple antigen binders, in fact. They can have other properties encoded into them. Half-life extension would be one example. We like to use llama VHs because they're small and highly stable, and we can move them around and we can build the bridging protein that we need for each specific indication that we're going after. Now, one key element is delivering these bridging proteins into the patient where the CARs are. So half-life extension would be appropriate for a bridging protein that we're developing as a biologic for injection. We work a lot in the lentiviral space, so in that sense, the bridging protein is made by the CAR T cell itself, secreted. We also work in the oncolytic virus space where the targeted tumor cell is transduced and it secretes the bridging protein that allows the CAR to then come in and attack the tumor cell. And I'll show you how that works. So bridging protein characteristics that we like is they're simple, they're modular, we can build them to be multi-antigen, and they're adaptable. And just to remind you, the anti-CD19 CAR T cells characteristics that we like are their persistence properties, their fitness properties, and their trafficking properties. So now we have to put these pieces together in order to build our platform technology. So bridging proteins, as shown here, consist of CD19 and an anti-tumor antigen binding domain, perhaps Her2, and binds to that Her2, painting the target tumor cell with CD19. It's pretty simple. You can see it in a FACS plot very readily. The tumor cell becomes bright for CD19. It follows that if you add an anti-CD19 CAR T cell, it will kill that CD19 painted tumor cell as if it were a CD19 positive cell, because the CAR doesn't care, it just sees the CD19. So the CAR T cell sees CD19, the tumor cells are killed. It's really that simple, and it works very well. Now, I mentioned that we can encode the bridging protein sequence into a lentiviral vector. That's illustrated here. We have a strong promoter, we have the CAR domain, a cleavage site, and then the secreted bridging protein. So this is an all-in-one solution. The CAR puts the anti-CD19 domain on the T cell surface. The bridging protein is secreted. We've got both of our pieces in play from a single transduction event. When that CAR T cell sees a B cell, it'll expand and proliferate just as any anti-CD19 CAR would do. When it sees a tumor cell that has the targeted antigen on it, the bridging protein will paint that tumor cell with CD19, and then we get local proliferation and activation at the site of the tumor. That's the vision. Toggling back and forth between the two sources of antigen. Activation, persistence on the one hand, local site of cytotoxicity against the tumor cells on the other hand. Now, how do we translate this clinically? So our first program is to tackle Her2-positive CNS metastases. The program's in IND enablement now. This is the same cartoon I just showed you. I've just replaced the words with an illustration of the bridging protein CD19 anti-Her2. This triggers potent cytotoxicity at subnanomolar concentrations. So that's an amount the T cell can secrete and achieve in vivo efficacy as illustrated here. This is a SCOV3 model, Her2-positive ovarian carcinoma, which we took out through day 40, by which time all the control mice were gone. 90% of the treated mice were cured. We kept those mice out another 15 days till day 55, looking for relapses. We didn't see any. So we said, all right, let's go back and forth between the antigens. Proof of concept, let's give them a CD19 positive tumor and see if they can switch antigens. And indeed they did. The cured mice eliminated the CD19 positive cell. We used the NOM6 because we can follow it with luciferase. These are naive control mice that were enrolled, and of course they developed a prominent systemic leukemia and had to be sacrificed quite quickly. So toggling back and forth between the antigens in vivo, if we do it in vitro, we found a very interesting effect. So if we re-stimulate CARs on a solid tumor cell repeatedly 4 times, 4 rounds of re-stimulation and resting, on a solid tumor cell we get modest CAR T cell proliferation, about 4-fold, 5-fold in this particular experiment. If we do a matched experiment with Raji cells, and we've mitomycin C treated the tumor cells so they can't proliferate and contribute anything to the assay. If we do the same exact experiment on Raji cells, we get 30, 40, 50, 60-fold proliferation of the CAR T cells. This seems to be a real biological difference between the embrace of a CAR T cell with a B cell line, Raji, versus the embrace of a CAR T cell with a solid tumor cell. And I think reflects um, immunologically relevant biology. We think we know what the mechanism is, and we'll share that with you at another time. However, the take home is that this result may further explain why CARs directed to solid tumor antigens don't fare very well. They're just not getting a productive embrace when they encounter the solid tumor cell. So back to HER2 positive metastases. I think you all know this is an area of huge unmet clinical need. Um, HER2 positivity is correlated with CNS Mets, and it's in part because the therapy for HER2 positive disease is an antibody therapy, dual therapy typically, um, and those antibodies don't cross the blood-brain barrier and cannot treat the CNS lesions. These patients tend to metastasize to the CNS, at which point they're really in serious trouble. So, the CNS is an interesting immunological space. We know that CAR T cells can get in. We know this from the CAR-19 literature on CNS leukemia and lymphoma. We also know that they can get back out because there's, a robust lymphatic drainage from the central nervous system, just as in any other organ. So our CAR T cells can go in systemically, expand in the periphery due to the provision of antigen on normal B cells. The activated CARs will traffic into the CNS where they will encounter HER2 only on the metastases. They'll attack those metastases via the bridging protein that they're secreting. And should they exit the CNS, they'll encounter B cells again in the periphery. So I think it's a very nice system for this particular technology to work well. It's also good for the patients and the clinicians. And the reason is that the technology has a unique advantage of uncoupling expansion and persistence in the CAR from the antigen on the tumor cell. That means that the patients can remain on standard of care therapy to control their systemic disease. They don't have to come off dual antibody therapy, TRAS-PER, or TDM1 therapy. None of those antibodies cross into the CNS, but our CARs can expand off of B cells peripherally, and they can get into the CNS. This is a very interesting therapeutic profile that I think is quite unique and not achievable with other therapeutics in this space. So we're anxious to move this one forward. Now, each program that we move forward spawns programs behind it because of the modular nature of the bridging proteins. Once we've made a module with one antigen binder, we can click on other antigen binders, and that's what's shown in the Aleta 003 program. I'm just going to give you a tiny bit of information. This program is being developed for the gliomas, broadly speaking. These are the stem cell-derived CNS tumors that plague pediatric and adult populations, share an interesting array of antigen expressions, allowing us to weave together dual and triple antigen targeting bridging proteins that we can then use in the treatment of these indications, with the backbone being our anti-CD19 CAR T cells. These will be CD19-based bridging proteins. So just to tease you a little bit with data, when we weave multiple antigen binders together in a bridging protein, we get synergistic potency. So these are single antigen binders, these are dual antigen binders, all on the same cell line that expresses both antigens. And as we move from single to dual antigen targeting, we move from 100 picomolar, very good, to 2.5 picomolar potency, extraordinary. So we get an antigen, antigen targeting shift in terms of potency. Importantly, our analysis of antigen expression patterns from the antigens we're pursuing for these diseases suggests safe targeting combinations that we can use in the lentiviral system. And if we move to the oncolytic viral system and do local delivery of the bridging protein, additional combinations become available to us. So it's quite an exciting space. So I want to leave you with our pipeline. We talked about the CNS metastasis program underway in IND enabling study. I gave you a little bit of info on the dual antigen and triple antigen targeting proteins programs. I did not talk about the antigen bridging biologics, and I'd refer you to my ASH talk from last year on the CD20 program. We've got a multiple myeloma program as well. The CD20 program well on its way toward the clinic. And at another time, we'll come back and update you on our AML program. So with that, I will say thank you and see you on the other side for questions.